So, linkage crossing over and recombination. So, let us discuss this. What is linkage, recombination, and linkage crossing over and recombination? As you know that genetic study, when Mendel was doing the experiment and he said so many things regarding, he revealed that some of the genes do not follow the law of independent assortment during gamete formation. What is according to the independent law of independent assortment? Each of the allele is separately getting assorted so that the characteristics which are not seen in the F1 generation, in the dihybrid cross we have seen, isn't it? We have taken round, isn't it? Round and wrinkled seeds, that is the shape of the seed, and color also we have taken it is yellow and green. But we have seen that it is independently getting assorted. It means that round is not getting mixed with the wrinkled, yellow is not getting mixed with the green, even though there are difference, that is a dihybrid cross, isn't it? So it means that every in the dihybrid cross it has to obey the law of independent assortment. But Mendel said that Mendel revealed one more thing during the gamete formation. Some of the genes will not follow the law of independent assortment. So this is why. What is the question there? Why it is not allowing not obeying the law of independent assortment? This is because the genes which are present on the same chromosomes. Because, for example, genes which are present on the, like in the last we have seen that the homologous chromosome, that is the genes are located, that is this gene like this it is located. But in this condition, so in that condition it is obeying the law of independent assortment. Separately it is getting assorted when there is a gamete formation. But here, the genes are all present on the same chromosome. Okay, so if the genes are present on the same chromosome during the gamete formation, this gene is transferring to the next generation. The gene is transferring to the next generation. So chromosome is transferring to the next generation. The set of all the genes present on this chromosome is transferring together as a single unit. Okay, I repeat once again, dear students here. See, according to the Mendel, the genes should the law of independent assortment. Each pair of allele is separately assorted and transferred to the next generation. But Mendel is only telling that in the process of linkage, he is explaining that there are some genes which are located on the same chromosomes. So here only they are located, isn't it? So when this chromosome during the gamete formation is transferred, isn't it applied? So these all genes, whatever is present in this chromosome as a single unit, it is transferring to the next generation. So in such condition, what happens, these genes which are situated on the same chromosomes are called as linked genes. What they are called as linked genes, isn't it? Because they are having very less gap within the two genes. You can see here the genes which are located on the chromosome are showing very less distance. So almost all genes are transferring to the next generation. Suppose if you just compare the two different chromosomes, isn't it? In one chromosomes, we are able to see the genes which are located, that is, as a single unit, most of the genes they are getting transferred to the next generation. Whereas if you see the one more the condition, that is the genes which are located on the another chromosome. These genes, when this chromosome is transferring to the next generation, all along the set of the unit of the genes is getting transferred to the next generation. What is going to happen here and here? Here, most of the characters of the parents, that is expressed by the pair of genes located on the chromosomes, there is a recombination, that is, the lesser chances of similarity and more chances of variations will be seen in that condition. Okay, now I have to understood. Once again, I can say here, according to the Mendel, he has said that some of the genes are not obeying the law of independent assortment during the process of gametegenesis. That is, haploid, only a pair of, among the pair of chromosome, only one pair is transferring to the next generation. That is, one pair is coming to the next cell, that is in the haploid gamete formation. Similarly, among the pair of gametes, only one gene is getting transferred into the gamete. But in some conditions, genes are present, all the genes are present on the same chromosome of one character. That is getting transferred, that is transferred to the next generation as a single unit. So genes which are situated on the same chromosomes are called as linked genes, and their pattern of inheritance is called as linkage. What exactly the linkage is? Linkage is nothing but that is a physical association of the genes which are transferring to the altogether transferring to the next generation. That is linkage, isn't it? So it has been also shown that that is by the Morgan, that is T.H. Morgan, during his studies on the fruit fly, he has studied, he did an experiment with the fruit fly, that is Drosophila melanogaster. What it is called as the Drosophila melanogaster. It is a scientific name of the fruit fly. Do you know what is a fruit fly? When you keep a banana, small flies will come, very small flies will be when the banana will start decaying, you can find more flies will be very small. Those are called as fruit flies. What those are called as? Fruit flies. Those are called as. Okay, so these fruit flies, if you see, okay, T.H. Morgan. We did an experiment with the fruit fly. Hence, it is he was said to be was working on the fruit fly. Flying lab used to be known as flying lab, isn't it? At Columbia, that is University of New York. Okay, University of the New York. Why he has selected only fruit fly? He could have selected the other also, isn't it? He was known for his work with the this fruit flies. He got a Nobel Prize also for that, and he noted that Drosophila melanogaster, it is showing, that is a study on the Drosophila, that is in a small tiny that is flies will be there. So these flies here, Drosophila melanogaster. Okay, on that it is a small tiny flies which are present. Usually you can see easily, you can get these flies when you keep a banana, when the banana will get overripe. Obviously you are going to find some of the small tiny flies will be moving near the banana, that is nothing but fruit fly. Scientific name of that is Drosophila melanogaster. His work made such a thing that he has gotten Nobel Prize, and he was used to known as a fly lab at Columbia University of New York. And here, this is said to be T.H. Morgan. He did many experiments. So many experiments on the fruit fly. By taking the fruit fly, he did a lot of experiments. So that is the reason he is also said to be father of experimental genetics. He is also said to be father of experimental genetics. Why? Why he is said to be father of experimental genetics? Because he did a lots of experiment on the fruit flies, and fruit flies are said to be Cinderella of genetics. What they are said to be? Cinderella of genetics. Their fruit flies are named as Cinderella of genetics. Isn't it? So why only he has selected the fruit flies? He could have selected other animals, other plants, or other animals, or anything he could have selected. He has selected. The reason we will come for the 3-mark question. Linkage. In the linkage, you can see the definitions will come. That is, what is linkage? The definition will come for the 1 mark, and apart from that, the types of the linkage, incomplete and complete, I will show you after this. I will explain you. That will come for the 1 or 2 mark, and this will be asked for 3 marks. Very repeated question in the PU board, and also relatedly very so many NEET and CAT questions will be arising from this part of the linkage. That is how the why, what are the reason why we have selected the Drosophila melanogaster. First it is easily availability. First reason is availability. Okay, availability. So we can easily get this fruit fly on a synthetic media. We can prepare it and keep in the lab, or else we can also keep a banana very after some time. Immediately when the banana will get overripe, immediately we can find some of the flies which are going to appear near that. Isn't it? Then the second reason is that is the short life of the Drosophila melanogaster. That is, the life cycle is short life cycle. Life cycle is very short, isn't it? It is within 10 to 12 days the life cycle, isn't it? So this is also one of the reason. First reason you can see here, that is it is easily available, and the second reason is it is very short lifespan, and you can see many of the generation within few period of time. Then apart from that, a single mating of the pair can produce hundreds of offspring. Single mating of a pair can produce hundreds of progeny. Hundreds of progeny. First reason why he has selected, reason for Morgan to select the Drosophila for his experiment is it is very easily availability is very much important. For example, if you select such a organism which you don't find, are you able to do the experiment? No, you cannot get the animal result. So easily availability is one of the reason. Second reason is life cycle is very short, and that is only within 10 to 12 days the life cycle is going to be ended, and single mating of a pair can give hundreds of progeny. So many progenies are available, means what happens? You can see the differences and similarities between the progeny. If you are crossing a parent with the another parent, so what are the progenies? In that you can see if there are only 1 or 2, you cannot see the similarities, and you can mark out many similarities and variations among that. If they are present more in number, that is hundreds together, so can easily mark out. And one of the reasons are they are showing sexual dimorphism is also there. Sexual dimorphism. Sexual dimorphism means you can easily identify the male and female. Male and female. That is also very much important to know that how many male progeny got the characteristics like the male parent, and how many female progeny got the characteristics like the female parent, isn't it? So that is the reason here you are able to see that sexual dimorphism is very clearly there. Like in the bisexual flower, Mendel selected the plant because of the bisexuality. Here also it is showing that, and altogether only 4 pairs of chromosomes are present. So one more thing, that is only 4 pairs of chromosomes are there, isn't it? Only 4 pairs of chromosomes are there. Only 4 pairs. Chromosomes. Okay, among that, you can see allosomes, that is XX and XY type of sex determination. We will be studying the sex determination, what exactly, isn't it? XX and XY type we are able to see there, isn't it? Similar to the human beings, that is XX will be the females, isn't it? In human beings also you can see, dear students, that is in the humans you can see that is totally 23 pairs of chromosomes are there, isn't it? Among that 22 pairs are autosomes. Autosomes means body characteristics which are also called as somatic chromosomes. Somatic chromosomes. Okay, it means that our hair color, eye color, shape of the eye, skin color, all the body characteristics are depending upon this 22 chromosomes, which are same in the male and the female. But the last pair, that is the last one pair, that is 23rd pair, that is called as, in the male it is 23rd pair, in the male is XY. 23rd pair in the female is XX. These are called as allosomes, which is determining the sex of the organism. If XY chromosome is present, that person will be the male. If XX is there, that person will be female. Similarly, in Drosophila also, XY means male. Drosophila, that is fruit fly. If it is XX chromosome is present in the allosome, that is female fly will be there. So that is, it is determining the sex of the organism, which is also said to be sex chromosome. That is determining the body character. Autosomes are determining the body characters, right? So they are said to be somatic chromosomes. Then here it is the last pair. So totally we are having 23 pairs better. Here only 4 pairs. If you study 24, all the pairs are going to be getting, isn't it? So we have selected the Drosophila because of this reason. This is very important. It will carry 3 marks in the theory board also, and also so many questions around this will be asked in the written CET. It is easily availability of the fruit fly, life cycle, isn't it? Life cycle is very short, only of 10 to 12 days. If life cycle, if we could have taken the animal whose life cycle is 60 years, means we have to wait for that much to see to mature it, to reproduce, to get the next generation, isn't it? Single mating of a pair. Once the male and female is mating, so that is giving more progeny, hundreds of progenies. So you can easily see the number of differences and similarities. Then sexual dimorphism, you can easily mark out which one is the male fly and which one is the female fly. That is sexual dimorphism. In the flower we say that we call it as bisexual flower. We say that is different. That is unisexual flower we will say. And here we are able to see that is dimorphism, both male and female separately, and only 4 pairs of chromosomes are present in this fly. Among that, 3 pairs are the autosomes and 1 pair is the allosome, that is a sex chromosome. Like in the human beings, here also the sex determination is XX and XY. XX means female and XY chromosome is present, means it is said to be the male chromosome. Okay. Okay, here we'll end. In the next class, we are going to talk about the continuation.